pleased to be here to speak with you. Hardly any of you will know me. I'm not a synthetic biologist. I'm a cell biologist working on the problem of diabetes. And I see the point of my talk here to be a little bit more than light entertainment. It's sort of to present a challenge of diabetes about how synthetic biologists might think about approaching that. And I'm going to focus my talk really on ways that you could think about treating diabetes, and I think you'll see there are 2 or 3 applications, or apps, as Nubar said, that might deserve some of your attention. So I'm going to begin by reminding you of stuff you see in the newspapers. I won't read these statistics to you so much as just to say that diabetes or metabolic disease is one of, if not the major healthcare risk facing Western countries. It is an increasing problem in India and China. It's estimated there'll be more than 120 million diabetics in China by the year 2040. Maybe one way to think about all of these numbers is to just reflect on how much insulin is sold. This year, $22 billion in insulin will be sold, and the cost of the US healthcare system of treating diabetes is estimated to be over $200 billion a year. So that's one way to think about there's a significant unmet medical need or a market. I'm going to summarize for those of you who don't think about diabetes, the 2 types of diabetes, just with some fun pictures of this. Here's a picture of what's happening to most Americans, an increasing number, that as we gain weight, we can be metabolically healthy, but overweight. And at that point in our life, I guess I'll focus here on this left screen, there's a normal number of pancreatic beta cells, the cells that produce insulin. As weight increases and insulin resistance appears, the pancreatic beta cells become overworked and die from high glucose levels. And my favorite real-life picture of this is not in a person, but is in a rat, a genetic rat model. And if you look at that rat on the right, you see that he's been feasting on a McDonald's diet for quite a while. And I don't know if the lights are too bright, but you can see here's his right side, and here's his left and his right sides. This is the fat content per pancreatic islet. And as that increases, pancreatic beta cells die. But initially, there's an increase in the number of beta cells to cope with the increased carbohydrate load. The increased carbohydrate load means that to just a quick review of basic metabolism, is as you eat food and you create in your digestive system sugar in the blood, that sugar cannot be taken up by cells without the hormone insulin. So in the absence of insulin, you can't make use of energy and you won't survive very long. Here you see that as the carbohydrate load goes up, the compensatory reaction is to make more beta cells initially, but this sort of dies out or poops out as the animal puts increasing stress on this. So this is what happens in the extreme of type 2 or adult onset diabetes. In type 1 diabetes, the same result is the loss of the pancreatic beta cell, but it occurs by a different mechanism, almost certainly caused initially by an autoimmune attack. So if you look here at this picture, and I'll come back to this picture again in a moment, there's a challenge for you. You can see in blue cells that are making the hormone insulin, and in other colors, cells that are making different hormones. So each cell in the pancreatic islet makes a different kind of hormone. In type 1 diabetes, the insulin-producing beta cells are destroyed, so the patient cannot make their own insulin. So I want to focus my 20-minute talk really on these blue cells, on the pancreatic beta cell, with the following argument. There's an increasing deficit in the number of beta cells. This is correlated with the increasing incidence of diabetes. Secondly, all genetic studies have in some ways surprisingly pointed to the pancreatic beta cell. When our colleagues here in Boston, David Altshuler and others, looked at GWAS studies for type 2 diabetes, it was largely expected that they would find genes in the periphery, in the muscle and the fat connected with insulin resistance. But in fact, all of the genes they found pointed to the beta cell as being the only association that they could find any statistical significance for at that time with type 2 diabetes. For type 1 diabetes, the GWAS studies point to 2 cell types, the T cells, the immune reaction, and again to the pancreatic beta cell. So my point here is that there aren't enough beta cells. Having more beta cells is better for your metabolism, how will we make more beta cells? And that's what I'm going to focus on in the next few minutes. So let's talk about making pancreatic beta cells. From a synthetic biologist perspective, and this is stretching it a bit, I would argue that what one wants to understand is how to instruct this cell as to which genes it should turn on and off. What is the sort of molecular biography of that cell as it moves from a cell that could make anything, an embryonic stem cell, to a cell that senses glucose and secretes insulin. So over the last decade, we've studied in quite enormous detail all of the steps that occur during normal ontogeny, during normal development, what genes come on and go off. And as you will appreciate, in a human embryonic stem cell or in an induced pluripotent stem cell, there are lots of genes that come on and go off at each of these 5 or 6 stages of the process. There are other ways one can think about making a pancreatic beta cell. I'm not going to talk about it today, but it's possible to discover, for example, new hormones that stimulate the synthesis of new beta cells in people that still have a residual number. And a third way would be to reprogram a pre-existing differentiated cell. So you could think about what cells in your body you might have excess of. Blood cells would be one, but they're not very long-lived. We've mostly focused on liver cells and intestinal endocrine cells to try to convert them into pancreatic beta cells. But I'm going to really focus in for this group on this first thing of the directed differentiation of embryonic stem cells. We have prosecuted this by making fluorescent reporters in these cells and studying the normal development in rodents, and then to the extent possible using human fetal material to confirm our findings on what genes come on and go off. I'm not going to show you any of that data, to just say if we know what genes have to come on and go off at each of these stages, it then allows you to look for growth factors or transcription factors, or in our case, we've been looking for small chemicals, small molecules that can instruct the cell as to what its next step in the process should be. And again, I'm not going to show you all of that data. I'm going to instead concentrate on the challenges. But just to say that we've gotten quite good at, I would say, the first 3 or 4 steps of this process. And I'll show you that we can make large numbers of cells, and then what is the challenge going forward. Just as a reminder then, the red cell here can make any part of the body. And the trick is to tell the cell that it should first become endoderm. For those of you that didn't do developmental biology, the endoderm is the embryological part that makes all the organs that run from your mouth to your anus. So your esophagus, your lung, your liver, your pancreas, your stomach, your intestine, all come from the endoderm. After you tell a cell to become endoderm, it then has to become pancreatic. The pancreas, I forgot to tell you, and I should go back to show you that slide. The pancreas is about the size of a banana. Most of it is exocrine tissue, which secretes enzymes into your gut to digest food. But within that banana, kind of peppered through it like raisins and raisin bread, are these islets. These islets are spheres of 1,000 to 10,000 cells. A mouse has 200 of these. You, if you have a normal dollop of them, have about 800,000. So within your pancreas, there are about 800,000 of these spheres, which means in total you have about 1 billion of the blue insulin-producing cells. Okay, how do we go about making them then? The advances in this area in the last year or so have been to begin with this challenge of trying to make the cells, but then thinking about scaling it up. How would we scale it up from 2-dimensional growth into 3-dimensional growth in these spinner cultures? And the point I want to make here is relatively straightforward, is that we can begin with single cell suspensions here, and there are a few clumps of cells. And then as these cells grow over a period here, within a few days we can make as many as 500 million cells in 300 milliliters. In other words, 2 of those devices, which are about this tall, will make enough cells in principle to treat 1 patient that doesn't have any pancreatic beta cells. The second thing that's been an advance is that by performing these differentiations in these so-called spinner cultures, or in 3 dimensions, we're able to get surprisingly homogeneous differentiation. If you look at the green cells there, they're being stained with a transcription factor called pancreatic duodenyl homeobox number 1 That transcription factor is expressed only in the cells that make the pancreas. And you can see that we get quite good uniform staining up until that point. And as I said, the main thing I wanted to emphasize here is that we can now get routine production, even undergraduates do it, of making 500 million cells in just about 3 weeks. So what are the challenges here? This is what the cells look like at the end stage now. So this is a fixed section through one of those spheres, and it's been stained for insulin. The insulin-stained cells are in brown. And so you can see there pretty well about half of the cells have done what we want them to do. So that brings up a point that I have not mentioned so far, which is at the end of the process, things go a bit awry. We don't actually understand yet how to tell the cell to make just pancreatic insulin-producing cells. We don't know what these other cell types are. They don't seem to be making other hormones. They could be stuck at an early stage. But we have what I would call a kind of homogeneity problem at the moment. And I think that's a solvable problem, but we've had this problem for more than a year. The second problem, not shown here and something I haven't mentioned yet, is that while those cells make insulin, they do not properly sense it. I was just in the session here on sensing and accurately reporting. This is a different kind of sensing. They have to sense glucose levels, as your body does, with an exquisite sensitivity, and then squirt out just the right amount of insulin. Those cells that have been produced so far will respond to glucose, but not with an accurate sensing mechanism, and they usually just dump their insulin on the first challenge with glucose. So several groups who've been working on this, including 2 biotech companies, are going forward with the plan to provide these progenitor cells to patients and with the hope that they would mature inside the patient to make a properly glucose-sensing insulin-secreting cell. I'm going to finish this part by showing you some recent results on how we think we may have solved the problem of figuring out how to turn the cells into a properly sensing machine. This has to do with a technique I'm not showing you, which involves fixing cells in a way that allows them to retain a very accurate representation of their messages, and then sorting those cells and then doing transcriptional arrays, in fact, and doing RNA sequencing. And we've identified these sort of standard signaling pathways which are important for the last step. We haven't completely sorted this out yet, but by using this little machine here, I don't even remember the name of it. I think it's called a Bioraptor from Beckman Coulter. We can add 8 different combinations of things to different wells at different times and do a kind of low-level multifactorial test on which combinations of agonists and agonists for these receptors are needed to move the cells forward. And for those of you who follow this field, this is a very positive result, which has taken us years to obtain. I'll just explain it to you here. Using 4 chemicals combined, we can turn these progenitor cells into a cell for the first time that in vitro responds to low and then high glucose at least twice. And so we haven't yet solved the problem, but I think we're now on the path to solving the problem of making, in short, 1 billion cells that accurately respond to glucose and secrete insulin. If that's true, it's still, let's say, a year away. The challenge then, I think, for a different aspect of sort of synthetic biology is to combine it with bioengineering in this challenge I would call encapsulation. Encapsulation has been identified as a problem for more than 2 decades. The problem being, in my opinion, that there has never been a reproducible, a regular source of cells for bioengineers to test their encapsulation devices with, because if they isolate islets from animals, they're highly variable. And some of the best work is being done here at MIT, but I would say even at that, there haven't been major advances in encapsulation. So particularly for some of the younger people in the audience, I show this slide to say what would be a good problem to solve is to create a device, and here I show you results where it's been held in a device for a few weeks, which allows for the survival of insulin-producing cells here in green, glucagon-producing cells here in red. These blue cells aren't doing what they're supposed to. We don't quite understand why they have lost hormone expression. But this device, so if there's, let's say, a membrane here, like a tea bag on either side, has to allow glucose in and allow hormones out, but importantly, cannot allow any cells in. And the devices that exist so far don't really have a problem with allowing cells in, but what they all do is attract fibroblasts. They become fibrotic very quickly, and they get essentially clogged up. It sounds like a simple problem, but it's a big problem. And if that could be solved in a generic way, it would not only be these kinds of cells, but other kinds of sensing devices could be put into people, I think, in a very interesting new approach. So the conclusion from the first part of my talk is simple, is that I think in the next year or two, it'll be possible to make virtually unlimited amounts of glucose-sensing, insulin-producing cells. The challenge will be how to get those into people to sense the body's glucose and squirt out the right amount of insulin or glucagon. I want to, out of fairness, present another approach to this problem, which I think is even closer to being put in practice. This is the idea, again, an old idea of an artificial pancreas or a closed loop system. It involves, on the left here, a continuous glucose sensor. It's an implantable device which reads blood glucose levels. And then it can talk to a pump which can secrete insulin and glucagon just in the right amounts to keep blood sugar levels at the right level. The challenge here has been to find good control algorithms. And recent work that may not be known to you, done here by Russell and Damiano, Russell at Mass General and Damiano at BU, have, I think, very impressive results, not on solving the problem, but on getting a long way towards it. So they've made an app, as Nubar said, on a cell phone-sized computer that talks to an insulin and glucagon pump. That is connected and reading a Bluetooth signal from a continuous glucose monitor. And what I'm showing you here from their paper is if you don't have diabetes or serious metabolic disease, your blood sugar levels are kept between a number measured in this old-fashioned units of milligrams per deciliter, but the numbers are just 80 to 120. So the green is the sort of right area you want to be in. This shows the blood glucose measurements of a person that's walking around with a continuous glucose monitor, doesn't have any normal beta cells providing insulin. And the person has a pump that squirts out glucagon and insulin, glucagon in red and insulin in blue. So I'll just show you an example here, that as blood sugar levels start to rise, so right here, probably after a meal, the pump is starting to squirt out insulin here in blue. If the blood sugar levels are starting to get too low, glucagon is squirted out to produce more, by gluconeogenesis, more glucose in the blood. Now, there are two ways to look at that. One is that there's a lot of excursions out of the normal range. The other way to think about it is that this is a completely artificial device that is now slowly coming to do what millions of years of evolution have allowed the pancreatic beta cell to do. The last thing I would say about this is some work initiated here by Peter So and Lauren Luger and Rich Lee at the Brigham. And they've done some very nice yet-to-be-published work with a slightly different idea on the sensoring side. The sensor I told you about in the previous slide is a needle that has to be implanted and kept into the patient. Here the idea is that they've taken green fluorescent protein, infused it with a glucose-binding protein, and now have very accurate readings on changes in fluorescence when glucose is bound. So their dream here is to put that kind of fused protein into a patient's cells. Here will be implanted, say, into the forearm, which would then read changes in fluorescence based on glucose, eliminating the need for another needle, giving a non-invasive glucose monitor, which would then talk to the insulin pump. So I appreciate that the two kinds of projects I've mentioned here as synthetic biology are quite far afield from what we heard about this morning. And I'm going to finish with even a more complicated one. And that is, how would you synthesize a system that would allow you to understand the degenerative disease? So we're not now talking about molecular sensors or cellular ones, but how would you combine different parts of a system to tell you what are the causes of a disease, what are the initiating events, and how does the disease progress? This problem, in my view, holds true for most of the major diseases from which everyone in this room or their loved one is likely to suffer from. That's all the neurodegenerative diseases, metabolic diseases. They're diseases which are multigenic and which have an unknown environmental factor that stimulates or initiates their cause. So I'm going to give you the example of doing this for type 1 diabetes. I've talked to you so far about using stem cells to make a kind of cell like a beta cell that you would think about putting back into the patient. But I'm now going to just finish with a disease modeling idea where you would use stem cells to make the cells that are involved in a disease and see if you can reconstruct it. This is, for type 1 diabetes, involves a requirement of a minimum of 3 different kinds of cells. The target cell, shown here in blue, is the pancreatic beta cell, which is killed by the immune system. The effector cell, or the killing cell, is a white blood cell, an activated T cell, which is educated in the thymic epithelium. So if you wanted to understand how this disease develops, you'd need to make hematopoietic stem cells that'll give rise to the T cells, and then figure out how to educate them as self or non-self, or as it occurs in this disease, improperly, to kill one's own resident beta cells. Where we stand on this project is to make the cells from a patient which has the disease, so-called diabetic IPS cells, or DIPS. So we've made these diabetic-induced pluripotent stem cells, and I've talked to you earlier here today about the progress in trying to get them to the beta cell. The plan for this grand reconstruction is this, that in addition to making beta cells, we'll make hematopoietic stem cells. This is being done by George Daly and Derek Rossi. And then we also need to make thymic epithelia, the educating cell. So this is a very big challenge. I've already told you how hard it is just to make this one, and now I'm upping the ante by making 3 types. But all of these together are going to be put into a living test tube, a NOD-SCID IL-2R gamma knockout mouse, essentially an immuno-incompetent mouse, prepared by Dale Greiner and Lenny Schultz. And then in terms of a grand synthesis, you could say, the idea is, can you watch human diabetes develop in this living test tube? And I will just finish by telling you why I think that's so important. At the moment, we don't know the answer to the question, if you cloned a person that had diabetes, would all of the clones get the disease? If they got the disease, would they get it in the same way and at the same time? In other words, we're completely ignorant about what triggers the cause of these diseases. So the idea here would be to take diabetic IPS cells from a patient and reconstruct the disease several times over and try to figure out, in the Aristotelian sense, what is the primary cause. So I'll end and be glad to take questions. Just note the people and collaborators here who've done this work in my lab. And thank you again for letting me come today. Thanks, Nabar.
Thank you. I have a few minutes for questions. Maybe I could go. Harvey, you may want to, let me get your mic.
I'll just shout. Can you talk about the immune problem, even if we made beta cells from IPS cells, would they not, for type 1 diabetes, will they not get rejected in the same way that the patient's own beta cells would?
Yeah, Harvey's pointing out something which I implied and didn't state and should have so explicitly, is if we make beta cells and put them into a type 1 diabetic, they will be rejected. There's a very good experiment that suggests that, done with taking pancreatic islets from a twin that didn't have the disease and transplanting them into their diabetic twin. And that's really why I emphasize this challenge of encapsulation. So to block cells from coming in or to block the immune cells. So for type 1 diabetics, making beta cells alone solves, I would say, half of the problem. For the tens of millions of pre-diabetic, yet type 2 diabetics, that wouldn't be a problem.
Doug, if I can add to that question, does it matter where these cells are in the body?
Yeah, that's a good question. The 2 general things we don't know is, one, are beta cells sufficient to actually treat the patient? We might need to make islets rather than the beta cells. And to your question, Nubard, where do the cells have to be? In some humans that have been tested, and certainly in rodents, they do not need to be in the pancreas. The human grafts of islets done by the so-called Edmonton protocol are all put in the liver, in the portal vein. And the evidence is that they survive there for at least 3 years now, and probably longer. The evidence from dogs with the encapsulation devices and monkeys is that at least for 18 months you can put them in a device and they'll survive and function. But as I indicated, the encapsulation devices suffer from this fibrotic reaction, which I think is the principal problem. Yeah, thank you.